So, good evening. And it's a huge pleasure to be here, this fantastic event, and especially after that extraordinary introduction. I really have something to live up to. So, I'm going to talk about my favorite subject, which is, can we live forever? And we've certainly been pretty good at making ourselves live for a lot longer. So this is illustrated here by a historical slide, and what's plotted is life expectancy at birth. So that's the length of time the average person is expected to live against the year in which the birth took place. And you can see that that covers 2 centuries. It started in 1840 and is projected through to 2040. And what you see is this extraordinary increase. So each spot represents the country that was the world leader at the time. So to start with, that tended to be the Scandinavian countries, various others in between. These days, it's East Asia. I think South Korea has the highest life expectancy for both men and women. And this is really an astonishing trend. It's 2.5 years per decade increase in life expectancy, driven by different things at different times. So to start with, it was infant mortality and childhood mortality, but then various public health measures, cleaner water, better food, better living conditions, immunization, antibiotics, all started to click in. And latterly, the increase is happening mainly in the older section of the population, and it's also fastest in medium income countries at the moment. And those horizontal lines were various people's predictions of where the trend was going to top out, what the intrinsic limit on our life expectancy would turn out to be. And as you can see, the trend has shot past all of them. We really don't know where this thing, or whether this thing, is going to top out. These are the recent figures for Denmark. So this is a similar plot, but for a 50-year period now, 1970 through to 2020. And the upper line is life expectancy for women, again from birth, and the lower for men. And you can see that here too, the trend is continuing. Women outlive men everywhere, in all countries in the world. But those extra years aren't particularly healthy years. Women have a longer period of disability and various diseases at the end of life than men do. But overall, I think we have to say this is a trend to be hugely celebrated. We've managed to keep ourselves healthier at a given age, and so we're living longer. We've really improved our living circumstances. And as a result, we're seeing some absolutely amazing lifespans. There are some real outliers, and these are 2 of them. So the lady on the left is a French woman who was born in the south of France in 1875. And she, as far as we know, is the world record holder. And she lived to about 122 and a half. I think we don't know why her. She came from quite a long-lived family, and there's a very small heritable element to these very long lives, but mainly it seems to be environmental rather than genetic. She certainly wasn't a particularly good advertisement for a healthy lifestyle. She did continue cycling until she was 100, but she didn't give up smoking until she was 117. So who knows what her secret was? The other thing is that there may be a more guilty secret involved here, because quite often it turns out with these records, especially the more historical ones, you have to be a little bit careful, because sometimes people had a motivation for lying about a family member dying, because if this was concealed from the authorities, they could continue to collect their pension. And there's a slight suspicion that she may actually have been her daughter rather than herself. But, and I think this remains unsettled. But be that as it may, there are still some fantastic records. So as far as I know, she's still the oldest living person today, Spanish lady born in 1900, and she's over 118 years old, so quite close to Jean-Louise Carmon. She attributes her success to having a happy and peaceful life, and also avoiding toxic people, which I think is quite a good bit of advice. So, something to be celebrated. But the downside is that we're clearly living much, much longer than we did even in the historical, let alone our evolutionary past. We're living to ages that haven't been molded by natural selection. It hasn't on the whole seen people as old as many of us get to these days. And the result is that healthy lifespan isn't keeping up with lifespan. There's a growing period of ill health at the end of life. And advancing age is the major risk factor for most diseases, actually, but also for the real big ones. So cardiovascular disease at the top, dementia at the bottom, in the middle, cancers at the bottom. And we've got 2 low, 2 middle, and 2 high-income countries in the column. And the 2 sexes, the beige is female and the green is male. And what you can see is no matter how you cut it, and there are obviously big differences in how much medical attention and diagnosis there is in these different contexts, but what's clear is you see this very steady increase in the incidence of these diseases with age. And because so many conditions are age-related, we start to see this, which is multimorbidity. So that's the presence of more than one medical condition in a single individual. So the plot here is different age categories along the bottom, starting at birth and going through to 85 plus. And then it's 100% up the y-axis, and it's the proportion of individuals that in pale haven't got any disease, and then 1, 2, 3, 4, 5, 6, 7, 8. And what you can see is as you cut it at later and later ages, you get a higher and higher proportion of the population with multiple diseases. And that's a problem for 2 reasons. First of all, it's difficult to treat. It's very complicated from a clinician's point of view. But the other thing is you start to get adverse interactions between the drugs that are used to treat different conditions. So it's a growing and global medical problem, this multimorbidity at the end of life. So what we're seeing is a situation where the major burden of disability and ill health now is falling on the older section of the population. So I think a very important question is, should we cure aging? And I would say that there's an ethical imperative to do so. People tend to think that somehow things going wrong with age is natural. It's just how things are. But if we think about illness at other ages, we wouldn't hesitate to intervene to try and improve people's health as they go through life. But I think there's quite an ageist attitude to the health of older people, and it's one that I think we should fight. I think there are things that we can do to keep people healthier for longer, to compress that period of ill health at the end of life. And that's actually what people like me who do the kind of research that I do, and there are quite a lot of us now, are trying to do. We're trying to improve people's health late in life. That might produce a slight increase in lifespan, but that's not the aim. That's happening already without the intervention of people like me. The aim is to deal with the problem that it's creating of this burden of late life illness. Now, it might seem rather naive, optimistic to say that we might be able to do something about late life health, but I think there are many indications that we can. And one of them is that the process isn't inevitable at all. So these rather simple creatures, the little hydra, freshwater hydra in the middle, dahlia anemone on the left, and this planarian worm, as far as anybody can see, as they go through time, they don't get any more likely to die, any less able to reproduce, any less able to do the sorts of things that they do. They just flatline. They don't show any sign of aging. And I think one of the interesting things about all of them is that they're basically bags of stem cells, these little undifferentiated cells that can divide and regenerate tissues. And these creatures have incredible powers of regeneration. You can often get a little bit of tissue from one of them and it will regenerate the whole animal. So it seems to be associated with this extraordinary ability of the stem cells to replace damaged tissues. But even if we start to think about creatures that are a bit more like ourselves and don't have these extraordinary powers of regeneration, we can still see some very long lives. These are just some of them out there in nature. So up on the left there, we've got bowhead whale, which can easily make it to 200 years. So they've actually been aged by the age of the harpoon heads that are sometimes found embedded in them, because nobody's tracked a whale for 200 years. So that's how they've figured out how long they live. And a few years ago, this Greenland shark appeared, on top right, and was discovered to live 400 years. In fact, it doesn't even start reproducing until it's 150. So extraordinarily slow life history. And then there's this little ocean quahog down on the left. It's just a very standard little bivalve mollusk, lives on the floor of the Atlantic. Turns out to be able to live 500 years. And I think we often have an intuitive idea that the reason the bowhead whale can live so long is because it's so big. It's something to do with being big. But actually, it's really clear that that association between body size and lifespan can be very easily broken during evolution. There's no causal connection between them. So we've got two quite small creatures down here on the bottom right, the naked mole rat. Fascinating animal. It's a rodent. They live in colonies in burrows with just two reproductives and the rest basically acting as slaves. They're intermediate between a mouse and a rat in size, but they can live about 30 years. And then that tiny Brandt's bat on the right, you can see it's just sitting on the tip of someone's finger, only weighs 8 grams, but can live 40 years. So we've got a rate of aging that clearly can evolve. And the differences between these species in how long they live is somehow genetically encoded. It's something to do with the genes in their genome. And so there's, of course, a lot of interest in can we find some of these genes, and can we find out how they work, and can we exploit that information to do something to help people. So in humans, there are some mutations that clearly affect the rate of aging, but they're very tragic ones in that they actually cause these premature aging syndromes, which happily are extremely rare. These mutants really, they're in the kind of dozen or 20 worldwide, but they do teach us something interesting about the aging process, I think. So the most extreme ones on the left, this is Hutchinson-Gilford progeria. So those children are born seeming completely normal, but starting at the age of about 18 months or 2 years, they start to develop the features that you can see, the loss of hair, the loss of subcutaneous fat. And gradually their health deteriorates, and eventually they usually die in their teens of cardiovascular disease. So a very, very rapid aging. Werner syndrome on the right is much milder. Again, the children seem completely normal when they're born, but in their teens, things start to go wrong. And again, they show signs of premature aging and eventually are very prone to both cardiovascular disease and cancer. And I think from learning about aging, the important feature of both of them is that they involve problems with handling the genetic material. So the mutation that causes Hutchinson-Gilford progeria actually affects a protein that's in the nuclear envelope in the cells. And the nuclear envelope keeps the genome arranged correctly, so the right bits are next to the right bits, and they talk to each other and they function correctly. But in these children, the nuclear envelopes start to break down, and so the genome is misarranged in the nucleus. And in Werner syndrome, it's actually a protein that's involved in the replication of the DNA. It's something called a helicase, so that there are errors when the DNA is copied. So I think that's telling us that fidelity, keeping the genetic material in order as life goes on, is very important for combating the effects of aging. But of course, what we'd like to do is not speed aging up. We'd like to slow it down to improve late life health. And for that, these creatures have really been the ones that have done the job and opened up the subject. So these are the standard laboratory model organisms. Budding yeast at the top, the nematode worm, Caenorhabditis elegans, Drosophila, and the mouse. And they're used because of the principle of evolutionary conservation. So, sorry, go back. What we can do is to use the simpler organisms, so particularly the worm and the fly, but sometimes also even bacteria, to understand the real basics. What's the genetic material? How does it work? How does metabolism work? How does the nervous system work? And you sort that out in the simpler organisms, and only then go and look at the more challenging and, of course, ethically more challenging mammalian situation. And that works because of the very powerful evolutionary conservation of biological mechanisms. I mean, those creatures may look very different, but many things inside them work very similarly. And you can often take a gene from one of them, or even from a human, and put it into another organism and find that it works perfectly well in the new environment. So there's a lot of evolutionary conservation. But I think people thought that principle might not work for aging. These creatures live very different lengths of time. So the worm does about 3 weeks, the fly about 3 months, and the mouse about 3 years. And they face very different kinds of challenges as they go through their lives. So why should they have common mechanisms of aging? And eventually someone who I think is really prescient just stopped thinking too much and just did the experiment. So what she did was to take the worm, feed them with a chemical mutagen which alters the genetic material in the progeny of the worm, collect the progeny and make strains from each of them, and ask, can we isolate a strain of worms that's long-lived? And what she found was that she could. And this was Cynthia Kenyon, working at UCSF in California. So what you can see here is the spectacular mutant that she isolated, which was called DAF2. And this is a survival curve now. So this is the proportion of the individuals left alive as time goes by. And the open boxes are the controls. So up to about a month, you can see this extraordinary increase in the mean and maximum lifespan of the mutant. And it took a while to figure out what this mutant was. Those were the days when cloning wasn't quite so easy. But eventually, it was found to encode the worm insulin-like growth factor receptor. So insulin, of course, much better known for its metabolic effects in mammals, its effects on blood glucose, and insulin-like growth factor for its role in growth and wound healing. And now here's a receptor turning up in the worm that's now clearly affecting the rate at which the worm ages. And this whole system is basically one for detecting good and bad times. The way that this receptor and the other bits of the network it's associated with work is to figure out what's the stress status of the animal, what's its nutritional status, and according to that, what can it afford to do? Can it grow? Can it reproduce? How should it regulate its metabolism to deal with the good and bad times? And if times are bad, if there's stress and a shortage of food, then the network shuts down and a whole load of proteins that it's been repressing become activated, go into the nucleus, and turn on genes that are important in stress response, defense, immune response, and so on, protective responses. And it seems that what this mutant does is to activate that whole system when times are fine. And very importantly, that extension of lifespan, it's not an increase in just the morbid period at the end of life. Those worms are wriggling around and active long after all the controls are dead. And the other very interesting thing about these mutants, because there's more than one of them, is that they can actually combat the effects of models of age-related disease in the worm. So one of the really early cases was a worm model of Alzheimer's disease, which is shown here. So what this poor worm has got is an expression of the A-beta peptide, which is associated with Alzheimer's disease in the brain of patients. It's thought to be in the causal pathway. And this worm is expressing the peptide in its muscles. And as you can see, it's in real trouble. Its back end is completely paralyzed, the front end just wriggling slightly feebly. But if you combine this with DAF2, we've again got the mutant worm on the right, you can see what happens on the left. We start to see a completely wild-type worm. It looks, for all intentions, just like an ordinary worm of the same age. It's wriggling around and perfectly normal. And this is true of a number of other disease models. So these are really quite powerful mutations that can improve the health of a normal worm and actually fight some of the causes of age-related disease. which is interesting, but of course we don't want a long-lived worm, we want a healthy human. So it's really important to know whether these effects are evolutionarily conserved. And happily, it turns out that they are. So first of all in Drosophila, and then in the mouse, very closely related, or mutants in very closely related genes, again turned out to be able to increase lifespan. And again in both of them, this was an increase in healthy lifespan, not the moribund period. So for instance, in that mouse, what you see is this really rather broad spectrum improvement across tissues. So they've got better glucose handling, even though this is the insulin signaling network, just tamping it down a bit actually makes them better when they're old. They've got a better immune profile, they've got better motor function, they're more agile and can hang on to a rotating rod, and they've got less osteoporosis. You can see the cataract there on the control mouse in its eye, and also there's ulcerative dermatitis on the head and nape. About 40% of the controls get that during aging. Never seen a case in the mutant mice. So it's a really broad spectrum protection. And again, they can protect against models of age-related disease, including Alzheimer's disease. So there are a lot of different mouse models of Alzheimer's disease, and there's been a lot of phenotyping done on them. And of course, one of the things we can look at is whether they have impaired memory. And this is an example where this has been done. So this is a Morris water maze. And it's used to test both formation of memory and testing it afterwards in the mice. So what you should be able to see, just here at the back, if I can get the pointer going, is there's a shadow. And what that shadow is in this cloudy water is a platform that the mouse can swim to. And although mice are good swimmers, they actually prefer not to. So if they've got something to stand on and stop swimming, they'll do it. So the mice are initially trained by putting them into the maze, and there's a flag on the platform. And so they swim around and eventually figure out that when they get to the flag, they've got a platform to stand on. And while they're doing that, they can also see those landmarks at the back of the maze. So they can learn the position of the platform relative to the landmarks. And then eventually we get to the memory testing phase, take the flag away, and look at the mouse, whether the mouse can remember where the platform was. And Alzheimer's mice are much worse at that than ordinary mice. They show an impaired memory. So here goes the mouse. So this is the Alzheimer's disease mouse. And you can see that it swims around more or less randomly. It shows no sign of knowing where the platform is. Whereas if we combine that with the mutant, what you see is the mouse swimming basically in the region of the platform, which it then finds quite quickly. I mean, obviously that's just an anecdote. These results are all quantified and statistically analyzed. And as well as dealing with the memory impairment, as the mice get older, they show poor motor coordination. At the stage where those experiments were done, they swim perfectly normally, but eventually they don't. That's rescued by the mutant. And you can see here the inflammation in the brain. So this is a region of the brain dissected from these mice. This is the Alzheimer's model paired with the mutant. This is, you can see quite a lot of the brain here. This is zooming in. And you can see this astrocytosis. So these are the astrocytes, the glia in the brain, and they become inflamed. They're stained for an inflammatory marker there. And you also get loss of nerve cells. And both of those are rescued by the mutant. So I think that's what makes these mutants so interesting. They're clearly making the animal healthier. So the main way that people have asked the question about humans is by looking at the human equivalent genes of the ones that have turned out to be important in animal aging. If you just do one of these standard, huge, genome-wide analyses, where you say, if we look across a whole huge populations and the whole genome, can we find anything that associates with lifespan? Actually, not much turns up. There doesn't seem to be a massive heritability. So people have taken the approach instead of saying, well, let's look at the genes that we know about from the animal studies. Do people who live to exceptionally old ages tend to have particular variants in those genes? And these are just some of the examples of the papers that have come out on that. These are just the headline publications. And you can see from the titles that we're seeing insulin, IGF, mTOR is another very critical bit of the network. And FOXO3A is important. I mentioned these proteins that become activated when the activity of the network is tamped down. FOXO3A is one of those. It's a transcription factor that goes into the nucleus and turns on the defense responses. And that's turned up, I think, in 7 separate studies now around the world as associated with survival to advanced ages. So I think more straws in the wind than anything hugely indicative of what's likely to happen with this network in people, but I think interesting nonetheless. So what can we do if we want to capitalize on this information to actually help people? Well, this signaling network that senses nutrients, it actually mediates the effect of an environmental intervention that's been known about for a lot longer, and one that potentially would be a lot more practical for humans, which is dietary restriction. So if you put an animal on a diet, you simply measure how much it eats if it's allowed to decide for itself, and then reduce its intake. You increase its lifespan. This has been discovered now across an enormous number of species. It was first discovered in rodents. And in rodents, you can really restrict them quite strongly. You can take them down to about 50% of what they would normally choose to eat. And basically, the harder you restrict them, the more you increase the lifespan. And it produces this really broad spectrum improvement in health. Just about anything that can go wrong during aging is rescued by dietary restriction. The only 2 things that they seem to be worse at are wound healing. So if there's any question of trauma, they have to be refed. And also they're not as good at clearing some viruses. Again, you have to refeed. And so because of this extraordinary, it's still the most powerful intervention that we have in the animal world, people have looked at what is the situation in rhesus monkeys. So these are 2 extremely long-term studies in the United States. The rhesus monkey lives about 40 years. And it was quite an interesting history of science thing almost, that 2 groups independently asked the same question: does dietary restriction extend lifespan and improve health in rhesus monkeys? But they actually implemented their studies in really rather different ways. And I think as a result, one of them found an increase in lifespan, the other didn't. They both found that they couldn't restrict the monkeys nearly as severely as the rodents. They could only go down to about 70%. But what they both found was an extraordinary improvement in health during aging. So these papers are still coming out, but these are just some of the health improvements that have been reported so far. So skeletal mass and physical activity are maintained. They've got better blood supply to both the heart and the brain, much better immune function, that's a really marked effect, decreased accumulation of iron in the brain, that's a bad thing that happens during aging, and less atrophy of the brain, less loss of nerve cells. They get less diabetes, less cancer, and less cardiovascular disease. So really a quite spectacular effect on health. So that's a nice indication, but there's a problem. Which is that when it comes to humans, I'm afraid most of us simply do not have the willpower to do dietary restriction. So there have been various attempts at clinical trials with dietary restriction in humans. There was one big one, and the aim was to reduce food intake only by 10%, so 90% of normal intake. Sorry, it was 80% over 2 years. And what was really noticeable was that there was a huge number of volunteers at the beginning of the study, thousands. They eventually, after various screening, got down to about 200 or 300. And most people couldn't comply. The best that anybody could do was 90%. And there was a big dropout rate. So as a public health intervention, it's completely impractical. It's just not going to happen. So I think the interesting question then becomes, well, can we somehow capture the beneficial effects of dietary restriction without dietary restriction itself? So for that reason, there's been a lot of interest in, what exactly is it about dietary restriction, just in terms of the food side of it, forgetting the molecular mechanisms for a moment, that's producing the effect. And it turns out, for starters, that it's not just calories. Actually, the composition of the diet is very important. So, for instance, salt, which is not really a nutrient, clear risk factor for cardiovascular disease, sugar for diabetes, and so on. But it also turns out that on the long term, in terms of health, protein is extremely important. So the basic story here is illustrated in this study, where what we've got at the top is the results of epidemiological analysis of self-reported food habits in humans. And at the bottom, we've got experimental data from mice. And what is clear from other studies is that you mustn't have too little protein in your diet, partly just for pure nutrition reasons, but also because it has a big effect on appetite. So it turns out that in both mice and humans, there's a kind of internally programmed need to take in a certain amount of protein over a given time. And if the diet contains too little protein, then what people and mice will do is to eat the diet till they get as much protein as the system demands, which will mean they overeat the other constituents of the diet to get there. So that's obesogenic. So very important not to have too little in the diet. But what this study showed is that it's also important not to have too much. So in humans, a high protein intake is associated, this is up to age 65, is associated with increased overall mortality and cancer mortality. So you want to keep protein down for that reason. But after about 66, and exactly when varies between different people, eating the same low non-cancer-inducing amount of protein as a young person does is associated with an increased risk of frailty, so muscle weakness and sarcopenia, and probably for that reason, an associated increase in mortality. So one size doesn't fit all in terms of age. The indications are that there should be a slight increase. But be that wrinkle as it may, it's clear that the protein content of the diet is very important. And that's really experimentally confirmed in the mice, where if you look at young adults, low protein indeed stops the progression of tumors, whereas as they get older, exactly the same amount of protein is associated with weight loss and muscle weakness. And there are other things about the diet. I can't tell the whole story, but it's clear that adjustment of the composition of the diet is something that could relatively easily be exploited to improve health. But the other thing that turns out to be important, and this is a very active area of research at the moment, all the answers aren't in, but it's when you eat. And this is mainly based on experimental work with mice. There's some data, and where there is, it's consistent, but there isn't much data on humans. So this story came out in this paper in Cell Metabolism, and it's a really very striking set of findings. So what they did was to take either genetically obese mice, so these are mice who don't have a leptin receptor, so they put on weight, or just ordinary wild-type mice with normal metabolism. And they subjected them to dietary regimes, either where they could snack whenever they liked, or where they were restricted to 9 hours food availability a day during the active period, which for mice is night, because they're nocturnal. And the results of doing that were really quite amazing. So what we have at the top is the obese mice, the genetically obese mice. And if they're allowed to just eat when they like, they become morbidly obese. And if you look at their metabolic profile, their blood glucose, circulating lipids and so on, they've got a terrible metabolic profile. But if they're time restricted, so they can only access food for 9 hours during the night, they don't eat any less. The total food consumption is the same. That's what's so striking. But they remain simply obese, and also with really quite good metabolic profiles. Basically the same story for the lean mice. Ad libitum, now they become obese with a bad profile, time restricted, they're fine. The bottom line, I've got no idea why they did this. They gave them the weekend off. my suspicion is that they may have been giving the experimenters a weekend off, but anyway, be that as it may, what we've got at the bottom is mice that were either ad libitum fed, or simply put on this time restriction from Monday to Friday, and then allowed to eat what they liked at the weekend. And they got as good a metabolic profile, and they remained as lean as the ones that were doing it all week. So I think this is telling us that there may be some very interesting things going on with timing of intake. Of course, if you can only eat for 9 hours, that means that for the other 15, you can't. So there's a long period of fasting. And that's true of dietary restricted animals generally, because what happens is the experimenters come in, give them their daily quotient of food, they eat it very quickly, and then they fast for the rest of the 24 hours. So it may actually be that fast that's more important than the restriction of the feeding. I think we need to understand more about mechanisms. But there's something interesting there. But also, as a result of parallel work in lots of labs around the world on aging, I think we understand its mechanisms much better, the underlying molecular and cellular mechanisms. And this has given us lots of ideas about potential targets. So the basic story is shown here, these hallmarks of aging. So the top 3 on the right, basic mechanisms of aging, are all to do with genetics, which I've already mentioned in the context of the progeria. So either instability of the genetic material itself, mutations and damage, the way it's packaged, the way it associates with other proteins, can all go wrong during aging so that gene expression becomes impaired. You then get things going wrong in cells, so they lose control of the protein domain, and of course those are the main effectors in cells. I've already talked about this nutrient sensing network. It seems to have evolved to drive at levels that work very well in youth, but are too strong in old age. So if you tamp it down, you get a benefit. Mitochondria are the energy powerhouses of the cell. They also have all sorts of other metabolic roles. They start to go wrong during aging. And then you start to see various cellular phenomena. So cellular senescence is important. Senescent cells are normally produced in the context of either development or wound healing because they remodel tissues. They're very biologically active cells that produce cytokines that can actually damage and remove the tissue around them, hence the importance in remodeling. But then when they've done their job, they're removed by the immune system. But that goes wrong during aging, and these senescent cells accumulate in tissues, and they cause damage. You can show in a mouse that if you remove them, either genetically or with a drug, that you can improve tissue health and actually extend the lifespan of the mouse. So they really are damaging. Stem cells, I've already mentioned in the context of Hydra and company. They can either fail to differentiate when they should, so they get a signal to please divide and regenerate the tissue, and they don't. This happens a lot in muscle. Or sometimes they just divide and proliferate when they shouldn't, for instance, in the intestine. So generally they become dysfunctional. And finally, we've got the systemic environment up at the top, left in green. So this is partly the immune system. There's a sort of sterile inflammation that happens during aging in almost all organisms. Not absolutely clear why or what its significance is. But there are also overall impairments to the systemic environment, the circulation, bad molecules in circulation. So all these things are happening. They're not independent of each other. They interact. So there's a complicated series of things that go wrong during aging. And I think the central thought in the whole field at the moment is right. We know that these mechanisms are involved in the aging process. They're all involved in the etiology of multiple age-related diseases. Let's tackle the underlying mechanisms of aging. Let's target them with drugs and try to prevent multiple age-related diseases simultaneously. So it's the prevention and the more than one disease that's the key here. This is the way the thinking is going in this field. Now that can sometimes produce a bit of a yuck response in people, partly because of this. There are a lot of remedies out there. If you go into any standard drugstore, you'll see stuff on the shelf for which there's no clinical evidence at all. None of it's been through a clinical trial. So there's a lot of fringe stuff around this work, which I think is problematic. But I think we also have plenty of exemplars of the sort of approach I'm talking about, where a lot of people take a drug for a long time to prevent something happening rather than because they've got a disease. And the really obvious one here are statins and the various different drugs that lower blood pressure. So they are taken by millions of people worldwide over long periods. Neither high blood pressure nor elevated blood lipids are diseases in themselves. They're risk factors for cardiovascular disease. But people take these drugs preventatively, they're very safe, and they've produced huge decreases in the incidence of cardiovascular disease. So I think there are perfectly sound preferences for the kind of approach that I'm talking about. But there are also big problems with de novo drug development for aging. So these drugs have been developed for specific conditions, the molecular basis of which is very well understood. If we think about aging instead, sorry, got my finger on the wrong button, there we go. What we're suggesting here is that aging is feeding into these different age-related conditions. So if we did a clinical trial with a new drug, we'd be looking at a lot of people over a long period and a lot of different clinical outputs. And I think that, honestly, the practical prospects of that are really rather low. I think the regulators would not be happy with it. The pharmaceutical industry would need to make money out of these very expensive clinical trials, and there would be big safety issues as well with a new drug being taken over a very long period. But, fortunately, it turns out that if we look at this, there we go, hallmarks of aging, actually, because they're all involved in the etiology of age-related diseases, there are already a lot of drugs out there that target various bits of them. So what there is a major prospect for repurposing of existing drugs, usually at lower doses, to actually deal with some of these problems that go wrong during aging. So if we think about the nutrient sensing network, which I've already been talking about, here it is blown up. This is actually the FLY network. Very simply, there are ligands, insulins, IGFs, a receptor, then a series of proteins that talk to each other, the signaling cascade, and then there's transcription factors that they control in or out of the nucleus. You can see FOXO over on the right. And I've just shown 3 existing licensed drugs that target different bits of the network. So on the left here, we've got this drug trametinib. It's actually used to treat malignant melanoma. And this bit of the network, the RAS pathway, is extremely important in cancer. There's a RAS mutation in about 1/3 of human cancers. So there are loads of drugs that target this pathway. This pathway is very important in metabolic disease. Again, lots of drugs. This is TOR, I've mentioned before, very key to sensing of amino acids. And there's a drug against it, rapamycin, and lithium targets this thing down here, GSK3. So what we've done as proof of principle is simply to look at whether these drugs are actually capable of extending the lifespan of flies, a nice quick test to ask whether in principle they might work. And it turns out that they do. So here we've got the survival curve of the control flies, each of these 3 drugs targeting their different bits of the network individually. We can also combine them in pairs because we know that they go for slightly different transcription factors, so they may be activating slightly different defense mechanisms. Sure enough, that's even more effective, these 3 here. And finally, if we put all 3 together, we get a stonking great increase in lifespan, something like 43%. So I think that's a strong proof of principle that we can mimic the effects of the genetic work that we've been doing with the drugs that target the molecules that we've identified from the genetics. And of these, rapamycin, I think, is the furthest on its way to actually being used clinically for protection against aging, because it was the first drug that was shown to extend lifespan in mice. So this is from the American Intervention Testing Program, and you can see here females on the left, 3 increasing doses, males on the right. You get a nice increase in lifespan, more in females than in males. Quite often these effects are sex-specific. It's an interesting drug. It was discovered on Easter Island. It's called rapamycin because the local name for the island is Rapa Nui. hence rapamycin. And actually an expedition went there to try and find soil bacteria that produced compounds that inhibited the division of mammalian cells. They were looking for cancer chemotherapeutics, and that's how they found rapamycin. And that is one of its clinical uses. It's also used to prevent tissue rejection after transplant. But armed with this information, quite a lot of small-scale clinical trials are happening in humans now. And I think this will be one way these repurposed drugs get in, by looking at specific indications. So humans respond rather badly, or old humans respond rather badly, to immunization against flu. They show a weak immune response, and they're less likely to avoid viral infections after being immunized. And Joan Manick, who led a company called Restore Bio, found that if she took older humans who were about to be immunized and either pre-treated them with not rapamycin itself, rapamycin's off license, but with a different drug, but with a very similar effect, then she could boost the immune response to the vaccine, and also there were fewer viral infections in the ensuing winter. So that's one way, I think, that these drugs will come in. And rapamycin is now being tested in the context of mild cognitive impairment and one or two other things. But they may, in the context of repurposing, we may also see actual trials for aging. And I think a very important development here now is that the FDA, the US organization that licenses clinical trials, has recognized aging as an outcome in trials. So I think there's about to be, if they can get the finance, a trial for metformin, which targets part of this nutrient sensing network. It's the drug that's now always used as the first defense against type 2 diabetes. It's taken by huge numbers of people. It's extremely safe. And it's a very good candidate, I think, for this kind of clinical trial. Further on the horizon, looking at some of those other hallmarks of aging, moving away from the insulin sensing network, cellular senescence, I mentioned, is very important in several diseases, particularly osteoarthritis. So there's an illustration on the left there of an arthritic knee. But similar in other arthritic joints, there are lots of senescent cells. And people are developing drugs now that can actually remove the senescent cells. This is shown on the right here. This is an experiment with the knee joint in mice, looking at whether a cocktail of chemicals can remove these senescent cells. The chemicals that have been developed are actually rather toxic. You wouldn't want to use them systemically. But a joint, a capsule, is an isolated context, as is the eye, where cellular senescence is also important in a lot of age-related eye diseases. So that's one thing that's going on. I mentioned the systemic environment goes down. There have been some really quite Frankenstein experiments with mice looking at this. So if you've got a single strain of mice in the lab, they're all essentially genetically identical. So you can conjoin their blood supply because there's no rejection, there's no problem with coagulation. And what people have done is to join a young to an old mouse with the appropriate controls, the young to young, the old to old. And a series of these studies have shown that the stem cells in different tissues of the old mouse become regenerated by that maneuver. It's not quite apart from the ethical aspects, it's not an ideal experimental design, because what happens with the young and old pair is, of course, the young mouse is very active. The old mouse wants to sit and sleep, but gets dragged around the cage by the young mouse. So it's not a very well-controlled experiment, and increasingly it's being done with plasma. But it's clear that either there's something rejuvenating in young blood or something bad in old blood, and if they can be identified, it should be possible to manipulate them. So I think metabolites are also going to be very important. There are startups piling into the whole research area. I came to a conference here in Copenhagen last year, and these are the companies that were just sponsoring that meeting. And that's not showing the big ones. Some of the big pharmas are involved. There's a new group called Altos who are looking at tissue regeneration, reprogramming of cells. The Saudi government have just put in a huge amount of money. There's money pouring into the area. So I think the answers will come in faster as a result of that. And I think it's very clear that the health economics of this approach, targeting aging to prevent disease, are really very favourable. People sometimes say, but hang on, if you keep people healthier as they age, maybe they'll live longer and the world will become more crowded. I think there are two things there. I think increases in lifespan as a result of this approach are likely to be modest. And the important thing is the health, not the lifespan. But also, the number of people in the world is overwhelmingly determined by birth rates and childhood survival, not by a few extra years in older people. So I think it's a global challenge on a par with climate change, and that we really should address it. So that's enough of the homily. To summarize, mechanisms of aging lead to age-related diseases, and they can be targeted with both lifestyle, I haven't had time to say anything about exercise, but that's also very important, and drugs. Targeting the nutrient sensing network with a cocktail of drugs can be very effective. There are emerging opportunities like removal of senescent cells and rejuvenation of circulating factors. And the big hope is that we're looking to a broad spectrum, preventative set of interventions for the diseases of aging, with benefits for both quality of life and economics of healthcare. And I'm going to stop there, and thank you very much for listening.
End of transmission.